Let's start with theories of enzyme action. The enzyme substrate complexing hypothesis, Michaelis-Menten theory, a significant advancement in the understanding of enzyme action. A significant advancement in the understanding of enzyme action was the formulation of the enzyme substrate complexing hypothesis by Michaelis and Menten in 1913. It holds that an enzyme molecule provides a specific site on its surface to which its substrate molecule binds itself. The reactive groups on the enzyme soon initiate chemical changes in the substrate. According to Michaelis-Menten hypothesis, the enzyme E first selectively reacts with substrate and then enters into a brief and intimate combination with it and finally forms an unstable and highly reactive enzyme substrate complex with a rate constant K1. ES or the enzyme substrate complex has two possible fates. It may dissociate into enzyme and substrate with a rate constant K2, or it may make the substrate molecule activated S star and reactive even at low temperatures. In the second instance, the substrate transforms to a product and the enzyme substrate complex becomes the enzyme product complex with the rate constant K3. This complex soon breaks up into enzyme and product. There is a reverse reaction. EP is equal to ES is a negligible or practically absent, so it is assumed that the product never reverts to the original substrate. The release of the P from the enzyme regenerates free unchanged enzyme, which may repeat the cycle with other substrate molecule. Michaelis-Menten theory can be represented by the following equation called Michaelis-Menten equation. E plus S gives ES, gives EP and enzyme plus product. The rate of E plus S transforming into enzyme substrate complex is K1, and the next stage, that is enzyme substrate complex transforming to enzyme product complex, is K3, but the rate at which enzyme substrate complex is reverting into the enzyme and substrate, that rate is K2. So K1, K2 and K3. Okay, here S is the substrate, E is the enzyme, ES is the enzyme substrate complex, EP is the enzyme product complex, P is the product, and K1, K2 and K3 are the velocity constants or rate constants of the respective reactions. According to this equation, E combines with S with the rate constant K1 and forms ES. Then ES dissociates either into E and S with the rate constant K1, K2, or to E and P with the rate constant K3. The formation of enzyme substrate complex. E plus S changing into ES is a fast and reversible reaction, and it attains equilibrium very rapidly. But the formation of the enzyme product complex and the breakdown of enzyme product to enzyme and product are slow and irreversible. According to the Michaelis-Menten theory, the process of enzyme catalysis is completed in 3 stages as follows: diffusion of the substrate to the enzyme and its stereospecific binding with the active site to form the enzyme substrate complex. According to the Michaelis-Menten theory, the process of enzyme catalysis is completed in 3 stages as follows: diffusion of the substrate to the enzyme and its stereospecific binding with the active site to form the enzyme substrate complex, conversion of the primary enzyme substrate complex to one or more activated enzyme substrate complex, and finally to an enzyme product complex, and the detachment of the reaction product from the active site of the enzyme and its diffusion to the medium. So, enzyme plus substrate forming enzyme substrate complex, then the activated complex will be forming enzyme substrate activated complex, then again activation is happening. After that, enzyme product complex is forming. Later, enzyme and product get dissociated. The enzyme is again reused in the reaction. The first stage is usually quick and short. It mostly depends on the concentration of the substrate in the medium and the rate of its diffusion. Enzyme substrate complexing occurs to weak non-covalent bonds such as hydrogen bonds, ionic bonds, van der Waals forces, or combination of some of this. At this step, the lowering of the level of activation energy is very vital. The second stage is very slow and its duration. Second stage in the sense conversion of the primary enzyme substrate complex to one or more activated enzyme substrate complex. Conversion of the primary enzyme substrate complex to one or more activated enzyme substrate complexes, and finally to the enzyme product complex. That is the second stage. The second stage is very slow and its duration depends upon the activation energy. This stage involves the loosening and breaking of the substrate bonds and making it new ones. This is due to the progressive increasing activation of the ES complex. Once the ES complex is formed, the physical properties of the enzyme like solubility, heat stability, etc. may change. The second stage may have several steps involving the formation and decay of reaction intermediates. When there are several steps in a reaction, the overall reaction rate is determined by the step that involves the highest activation energy. That's a new point. When there are several steps in a reaction, the overall reaction rate is determined by the step that involves the highest activation energy. Such a step is therefore called rate limiting step. The rate limiting step will be the step which requires the high activation energy. In some cases, there will be more than one rate limiting steps. The third stage in the sense detachment of the reaction product from the active site of the enzyme and its diffusion to the medium. Detachment of the reaction product. Detachment of the reaction product. Detachment of the reaction product from the active site of the enzyme and its diffusion to the medium. And due to that, third stage is also slow and short. What is happening in the third stage? Detachment of the reaction product from the active site of the enzyme and its diffusion to the medium. So third stage is also very slow and short. It depends mainly on the concentration of the enzyme substrate and the rate of the diffusion of the product to the environment. So there are different kinds of bonds between enzymes and substrates. So in the later stage, we are going to learn about the bond structure and what are the bonds associated with the enzyme substrate, enzyme product, or enzyme substrate product complex, whatever it is. So that is the bonding of a substrate molecule with the active site of an enzyme takes place by weak non-covalent bonds such as electrostatic bonds. Why is the bond is non-covalent here? That's because when the covalent bond is going to create in between the site between the enzyme and substrate in the active site region, that bond will become very stable and it's an unbreakable type bond. So there won't be any further reaction, like enzyme will be stopping the reaction just by binding with the substrate. So there won't be no later stage productions of other reactions. The bonding of a substrate molecule with the active site of an enzyme takes place by weak non-covalent bonds such as electrostatic bonds, hydrogen bonds, and van der Waals forces, or a combination of them. Apart from these bonds, water also has a significant role in enzyme-substrate interaction. Covalent bonding never occurs between enzyme and substrate. Electrostatic bonding takes place between the oppositely charged groups on enzyme and substrate. This force of attraction can be explained by Coulomb's law, where capital F is equal to Q1 Q2 by R square capital D. Here Q1 and Q2 are the charges on the oppositely charged groups of enzymes and substrate. R is the distance between the two groups and D is the dielectric constant of the medium. F is equal to Q1 Q2 by R square D. R is the distance between the two groups and D is the dielectric constant of the medium. Here Q1 and Q2 are the charges on the oppositely charged groups of enzymes and substrate. R is the distance between the two groups and D is the dielectric constant of the medium. So electrostatic attraction varies between medium. It is weakest in water and strongest in vacuum. Now, the hydrogen bonding is quite common in enzyme substrate interaction. Hydrogen bond is weaker than covalent bonds but stronger than van der Waals forces. Van der Waals forces are extremely weak interactions. They are not specific attraction forces in the sense that they can occur between any closely placed atoms. Theories of enzyme substrate interaction. Now we are going to learn about the major theories associated with the enzyme substrate interaction. Two hypotheses have been proposed to explain the conformation of the active site and the physical mechanism of enzyme substrate complexing. They are the lock and key hypothesis, or the template theory proposed by M.L. Fisher in 1894, and the induced fit hypothesis proposed by Daniel Koshland Jr. in 1962. So first we will be learning about the lock and key hypothesis. Lock and key hypothesis is the concept that there exists strict structural complementarity and absolute specificity between a substrate and an active site of its enzyme. According to this hypothesis, an enzyme is a rigid template. Enzyme is a rigid template. Enzyme is a rigid template with a pre-shaped active site to which the substrate can bind. Rigid template with a pre-shaped active site to which the substrate can bind. It also envisages that the active site of the enzyme is a permanent or fixed part with two components, one for substrate binding and the other for catalysis. The binding site is an exact replica of its substrate in size and shape, so substrates with different conformations will not be able to bind with the enzyme. Thus, the substrate can be taken as a key and the active site of the enzyme as a lock. Only a specific substrate can bind with a particular enzyme, just as only one type of key can exactly fit into a particular lock. So, lock and key method of substrates will be there. Matching active site will be there. So, enzyme binds with that. So, if induced fit method is something which we are going to learn later. So mismatching active site can happen, but still that enzyme substrate complex is forming. Lock and key hypothesis accounts for extreme enzyme substrate specificity. It holds that the rigidity of the enzyme causes the repulsion or exclusion of the molecules that are sterically different from the actual substrate. Lock and key hypothesis accounts for extreme enzyme substrate specificity. It holds that the rigidity of the enzyme causes the repulsion or exclusion of the molecule that are sterically different from the actual substrate. Lock and key hypothesis appears to be very simple explanation for enzyme substrate complexing and absolute enzyme substrate specificity. However, it does not account for the flexibility of the enzyme, effect of the allosteric modulators, and the relative specificity in which several enzymes with different configurations can bind with the same substrate. The induced fit hypothesis appear to resolve the shortcomings. So that is what is termed as a lock and key hypothesis by M.L. Fisher in 1894. The next theory of enzyme substrate interaction is induced fit hypothesis. Induced fit hypothesis is the concept that conformational changes may take place at the active site when substrate comes in contact with the enzyme. This hypothesis enjoys experimental support. The cardinal points of the induced fit hypothesis are the following. Enzyme is a flexible and elastic molecule rather than a rigid structure. The conformation of an enzyme. So, enzyme is a flexible and elastic molecule rather than a rigid structure. The conformation of an enzyme or its active site may undergo alteration on contact with the substrate. Active site need not be exactly matching with the substrate. A precise orientation of the functional groups of the enzymes is necessary for enzyme substrate complexing. During enzyme action, there occurs a stereospecific initial binding. During an enzyme action, there occurs a stereospecific initial binding between the substrate and the active site of the enzyme. This is called primary binding. What is primary binding? During enzyme action, there occurs a stereospecific initial binding between the substrate and the active site of the enzyme. That is called primary binding. Contacting or stereospecific primary binding of the substrate with the active site induces the reorientation of the functional groups. When a substrate analogy happens to come to contact with the enzyme, contacting or stereospecific primary binding of the substrate with the active site induces The reorientation of the functional groups. This causes conformational changes in the enzyme and the formation of a matching platform for the attachment of the substrate. Consequently, there occurs a secondary binding of the substrate with the active site of the enzyme. This completes enzyme substrate complex. Before moving on to the details, first let's learn about there are two types of binding occurring. That is the primary binding and the secondary binding. Primary binding and secondary binding. What is primary binding? During enzyme action, there occurs a stereospecific initial binding. Stereospecific initial binding between the substrate and the active site of the enzyme. That is a type of stereospecific initial binding. So that is termed as a primary binding. The next one, contacting or stereospecific primary binding of the substrate with the active site induces the reorientation of the functional groups. This causes conformational changes in the enzyme and the formation of a matching platform for the attachment of the substrate. Consequently, there occurs the secondary binding of the substrate with the active site of the enzyme. This completes enzyme substrate complexing. So there will be a primary binding and a secondary binding. When a substrate analog happens to come into contact with the enzyme, some structural changes may take place in the enzyme, but the required disorientation and reorientation of the functional groups will not occur. So, when a substrate analog happens to come into close contact with an enzyme, some structural changes may take place in the enzyme, but the reorientation of the functional groups will not occur. So the proper enzyme substrate complexing and the subsequent reaction will not occur when a substrate analog happens to come into contact with an enzyme. Some structural changes may take place in the enzyme, but the required reorientation of the functional groups will not occur. That is, the secondary binding process will not be occurring. The secondary binding process will not be occurring. So, the proper enzyme substrate complexing and the subsequent reaction will not occur. According to the induced fit hypothesis, that is one we are discussing now, the active site of the enzyme is not initially a rigid replica of the substrate. Instead, it is flexible and elastic and can be appropriately modified by an incoming substrate. It can be modified by an substrate. Instead, it is flexible and elastic and can be appropriately modified by an incoming substrate. Thus, contact with the substrate induces the active site to undergo conformational changes. The contact with the substrate induces the active site to undergo conformational changes and also to adopt to a suitable configuration for substrate binding. The induced conformational change in the enzyme proposes and imposes some strain on the substrate. This strain induces the substrate to transforms to a product by substrate strain theory. What is a new point here? Contact with the substrate induces the active site to undergo conformational changes and also to adopt a suitable configuration for substrate binding. The induced conformational change in the enzyme imposes some strain on the substrate. This strain induces the substrate to transform to a product. Substrate strain theory. Now, which are the factors influencing the velocity of enzyme action? The measurement of the velocity or rate of an enzyme action is known as a turnover number or kinetic constant. The measurement of the velocity or rate of enzyme action is known as turnover number or kinetic constant. It denotes the number of substrate molecules transformed into product molecules by a single enzyme molecule in 1 minute. Now, we are going to learn another major topic, that is the factors influencing the velocity of enzyme action. The measurement of velocity or rate of an enzyme action is known as turnover number or kinetic constant. It denotes the number of substrate molecules transformed into product molecules by a single enzyme molecule in 1 minute. Quite a number of factors influence the turnover number or the efficiency or velocity of enzyme action. They include temperature, hydrogen ion concentration or the pH, of the medium, concentration of the substrate, concentration of the enzyme, concentration of the product, action of regulators, etc. All this are the factors influencing the velocity of enzyme action, which can be easily represented by a turnover number. Turnover number is nothing else but the number of substrate molecules transformed into product molecules by a single enzyme molecule in 1 minute. So that can be represented as a turnover number. That is something which determines the potential of enzyme. First factor is the effect of temperature. Enzyme catalyzed reactions are sensitive to changes in temperature, unlike the velocity and rate of enzyme action increase with increase in the temperature of the medium. This is because rise in temperature increases the average kinetic energy and the reaction velocity of the enzyme molecules. The temperature at which the rate of enzyme action is maximal is called optimum temperature. Up to the optimum temperature, the rate of enzyme action usually doubles with every 10 degrees Celsius rise in temperature, but beyond the optimum limit, it drops to zero. The optimal temperature ranges for animal enzymes is about in between 20 degrees Celsius and 45 degrees Celsius. For plant enzyme, it is between 40 degrees Celsius and 60 degrees Celsius. An extreme temperature beyond this will inactivate or destroy the enzymes. Certain animal enzymes are denatured at temperatures around 40 degrees Celsius, but most others are denatured around 50 degrees Celsius. Extremely low temperature also is destructive to enzyme. A number of enzymes are inactivated on cooling and are destroyed at temperatures close to 0 degrees Celsius. Even though most enzymes are thermolabile, some are thermostable. Adenylate kinase Adenylate kinase is an example. It can withstand short-term exposure to 100 degrees Celsius without losing its activity. The enzymes of the certain thermophilic bacteria, especially residents of hot springs, have high thermal stability. They can tolerate temperatures around 100 degrees Celsius or more. So we will be representing a graph with rate of reaction on the y-axis, temperature on the x-axis, and then rise in temperature, increase in reaction rate. Then after reaching an optimum temperature level, fall in temperature, loss of catalytic activity, denaturation of enzymes. First point is denaturation is happening. Okay.